start, we're going to be talking, starting the big thing today, metabolism. It'll be a slow start, don't worry. I wanted to cover before that, really quickly, some that came up. I told you guys that the order of a reaction is always measured, has to be measured in a lab. Just remember that. You'll often be confused because when you have a reaction, that has these coefficients, sometimes it'll seem like when you did the kinetics, it was always this, right? You were taking these. That is not necessarily the case. This is not equal to this, okay? The little a's and b's and c's up here, the coefficients, that's worked out by how many atoms have to be on the left and the right for those compounds to create one another. The kinetics are different. And so you'll be told them experimentally. They sometimes coincide with those coefficients, so it can be confusing. So that was all there. All right. So I showed you this metabolic map before and where things are. Today, where the different cycles are happening, today you are going, let me just make sure I'm casting, yep. Today you are going to be seeing this flow diagram more. It's the one that Dr. Gardner and Dr. LaFontaine use more, so I use it in most of my lectures too. So what are we going to be doing today? We're going to be looking basically at glucose, coming in here to glucose 6-phosphate, and then we're going to follow the arrows, okay? We're going to talk about mainly one disease state where the problem is getting this glucose out, and it's basically going to be a day of Le Chatelier's principle, meaning if you build up reactant, you flow down the arrow the other way, okay? But we're going to start to make it clinical. So there will be a clinical case, and I'll show you presentations, and then we'll work through this and say, why did those presentations happen? All right. Also, I will note this is the single lecture that I most urge students to maybe not use my online notes. So they're here, if you want them, and they're in all the normal places. Get rid of this. The only reason I say that is because this lecture, I've told you, there's a hidden link there, you know where they all are. But the reason I say that is because these online notes are a bunch of lectures because metabolism is this beast, and so there's just a ton there. So if you want to use them, great, but just click through on the hyperlinks to what you want. Just a show of hands, who's printing off those online notes? Okay, that was a tentative yes. Okay, it's fine if you do, great if you can use it online, because it's really, I know they're verbose. Okay, I think you'll definitely have to have all of these. The only objective I added this year is this top one, and I don't even think I put it on Canvas before then. Let me know if it's not there, but I'm quite sure it is. And again, like with all my lectures now, there's no assigned reading. I have some optional reading here and what I've covered if you want to look at other resources. All right, so why is metabolism important and what is actionable? I'm a firm believer in the science behind there's a lot that we can do before a patient should even get to you as a doctor, and when the patient comes to you, a lot of what you can do in that quality sense, quality adjusted life years, is actionable things on their end that are very cheap, right? So let's look at some food. So here we're looking at things that cause harm, and on the left side is the mortality due to them in bars, okay? And these different bars are basically how it kind of advanced a country is. Is it the United States? Is it sub-Saharan Africa? And so the orange bar is us, right? And then you go down to more impoverished countries. And so as you go down, I want you to look at the magnitude, right? So salt is this amazing cause of mortality, okay? Think about that bar, okay? And this is measures of hundreds of thousands of people. Think about this bar compared to down here, sausage, highly processed meat, which is definitely not good for you, right? There's been a lot of press about that in the last few years. It causes cancer. That is bad. But look at this bar, this orange bar, compared to that orange bar, okay? I find it very unlikely that physicians, when they talk to patients about a healthy lifestyle, are weighting the salt proportionately to the processed meat, right? And there is kind of, the salt is a mixed story. Like there's some people that respond to salt much worse than others, but basically it causes hypertension. And so on the right side here, you see how that can cause death. So the big red bars for most of these is cardiovascular disease, right? If all these things make our heart have a problem, oxygen doesn't get the brain, we have problems. Going down, and so this was that first objective that I said I added. Remember my rule about like when I require you to know stuff and not just look it up? It's when it's either very small and constant or you need to use it quickly, right? And so here's an example of all your patients are going to be interacting with food, and I want you to have a rough idea of these, okay? So I said in the objective, rank order the importance of these, and FYI, in the objective, I put them in the right order for you, so from most important to least. And I'm not gonna ask you to differentiate these two. I'm going to, if I asked you, to know that salt is much more important than processed meat or red meat, right? So the extremes, okay? So just so you hear it, if you're running and in the class, so you can fire the neurons. So salt is very bad. The next down is a diet low in whole grains. So I boxed everything right, green. These things are good. So the death is caused by when you don't have them. Over here are the red things on the right. Then after grains are diets low in fruit, which interestingly, look, diet low in fruit was significantly more important than diets low in vegetable. That would not honestly have been my intuition, right? But if you look at the number of deaths from cardiovascular, the fruit was very important. Diets low in seed and nuts. A report came out this week about basically having a third of a cup of nuts lowers weight. And so basically the nuts create a sense of fullness, right? Now that is not the same as going to Costco and housing a bucket while you watch Netflix. But if you have a little bit, it basically causes you to eat less calories overall, probably. So after nuts, you get to vegetables. Then we've already talked about the fats, the good fats in fish. Fish in general are here, interestingly, because then they split out omega fats lower. After fish comes fiber. I've talked to you about fiber, right? We're starting to hit some things again and again. Then the polyunsaturated fatty acids, legumes. Then down here, trans fats. Trans fats are awful. And luckily, we're phasing them out in processed food. Remember, this is Crisco. This is things that sit on the shelf that never rot, so there's a huge incentive for industry to use them. Not good for us. Then calcium, sugar-sweetened beverages. A lot of hype about sugar sodas, which, again, are awful. But when you think of the magnitude of effect, they're not as important. And then processed meat, milk, and red meat. The milk is the one that I would put an asterisk next to. I've talked to you about that because milk here is good because they want you to have calcium and vitamin D, but I think it's a lot of unneeded extra calories, and there's a lot of research on basically the milk lobby pushing that on schools and blah, blah, blah. Okay, so over here, I won't go through that. That's more information from the same article if you want to see it in different bar graphs that basically show you where you want to be and where the reference range is. And so these green things are good, and you want to be above this dashed line, okay? And I've I compressed the images so the file wasn't large for you, but it hurt here. So there is a link to the article if you want the high-res image. But in short, I boxed North America. So if you look at Arbor, you'll see times on the top green box here where this bar doesn't hit that red line, we're probably not getting as much as we should. And then down here are bad things. And so if our red line goes past this green bar, it's not good. Actually, the dashed line down here is the average of all globally. So that might be the same here. Yes, sorry. The dashed line is the average globally, and the green line is where we want to be. So see how globally we're all lower than where we want to be, okay? Down here, it's the same. The dashed line is global, and the green line is we want to be below that because these are bad things. Whoops. So interestingly, for example, salt, right, the big bad one, where are we? The U.S., North America, is just below this dashed line of the global average, right? What I found interesting is this is East Asia, right? They use a lot of soy sauce. They're the only ones above the global average. So they're pulling everyone globally higher. So I assume they have a lot of deaths because of that massive salt intake. Okay. Kind of holistic, but there's a lot of magnitude of effect change here, right? All right. Going on to show how we're doing in Indiana, here is the adult obesity rate heat mapped on the US. Okay, this is 2013. Here are all the states. I'm going to click through and you're going to see time change. Okay, so here's 2013. Here we are. If you look at this range, purple's the worst. This is over 35% are obese. Then 30, right? It goes down, so blue's the best. There's no blue. Okay, so starting at 2013, now we're at 2014. You'll start to see kind of darkening colors here, 2016, 2017, and 2018. And so we're in this region of the country that has large obesity, right? And it's kind of spread up towards us. Here's Indiana, and we are 15th worst in the country. So not the worst, but if trends continue, you would imagine that purple wave, you know, not in a political sense anymore, spreading up into Indiana. So you're a part of the solution. What do you do for that? Here is looking at things that you can actually do, okay? And there's some clubs that I've been in touch with that may try to partner with IUPUI to write legislation for the state to give them to try to address this. So if you want to be part of that, I'll put you in touch with them. Let me know. So here are examples of laws that different states have, and all of these laws Indiana does not have. So here are things you can do in the school, okay? Do you have safe routes to school? And this is the number of states that have that. Codified in law that they have to have that, right? So 16 states pay to have infrastructure so kids can bike to school. Do you have PE time requirement in elementary schools? 18 states do. We actually, there's another metric of do you require PE in school? Indiana meets that one. We do require it. I don't know if I've told you this personal scar tissue from my life, but my kid up in Carmel has recess that is 15 minutes long. So someone checked the box of we want PE, but they didn't have a PE time requirement. So you end up with a 15-minute recess, right? And you can have that for elementary school, high school, middle school. So that's increasing physical activity. Then you can do food quality stuff, early childhood versus community. So these are all known solutions that are targetable that Indiana's currently not doing, right? Defined physical activity, nutrition standards, preemptive stuff for the community, food security. So these are all things that are out there. So some of these half the states are doing now, and we're not in that. Notice there's no objective here. Okay, I'm not testing you. Okay, turn and talk to each other. Can you guys, don't look at your next slide, okay? Honestly answer, oh man, is this not casting? I'll try to get this going in a second. It was casting? Yeah. Okay, I think that one might have been lucky, we'll see. All right, 10 seconds. This will make me so happy. And the answer is, that's all right. You guys are going to be better than most in a second. Ready? Right. So where does it go? Settle down. All right. Ready? So you guys basically picked energy and heat. You did pretty good. CO2 and H2O is where it goes. I'll show you that. Ready? I appreciate whoever wasted their vote on that. And then feces. All right, so where do you stack up? This shows they asked this question to family doctors, dietitians, and personal trainers. Almost everyone said energy and heat. Only a few of the dietitians got the right answer. So I want this class, when you go out, boom, you are the docs that, who are these marrying docs that know where it goes, right? So we have fat. If you take 10 kilograms of it, it gets converted into carbon dioxide and water. So you are literally breathing out your fat, okay? How is this like, now, in a true scientific sense, this is very ivory towerish kind of, but the carbon dioxide and the oxygen are of a lower energy state than the fat. So you did create some heat in your body, but if you were like looking at a pie wedge of how much energy is in an atom, almost all of it is in the carbon dioxide and oxygen, right? You're not doing fusion still. So anyway, it goes out of your body. The atoms go out of your body, some through urine or sweat in water, and mostly through carbon dioxide. So how's that applicable? If you're a doctor and you think this is the case, in your mind, you're probably waiting, you need to run more, you need to get hot more. That's somehow, not ether, but that's somehow how this fat is leaving your body, right? But it's not. It's when you exhale and water, and that gets back to this big take-home of diet, where basically it's very hard to exercise your way into nutrition. Really, you need to lower the calories coming in, okay? And then you just let your resting metabolic rate breathe out the fat because you're not taking in as much. All right, again, no objective, just make me proud. All right, so if you downloaded the PowerPoint yesterday, I think the only difference you'll see is I had these two emoticons wrong. But so I'm going to use these emoticons on this slide and the next to talk about what's happening to glucose. It's coming into the cell, it's going out of the cell, you're adding it to glycogen, then you're branching or debranching it from glycogen, and then you're taking, and then you could be taking it away from glycogen, right? So here's glucose. You get it into the cell. At all of these points, if you have a break point, like a genetic bottleneck, there's a problem, and we're going to talk about a few of them. So you get it into the cell, then you're adding it into glycogen. There's branching and debranching enzymes. All of that can go wrong. Then you have to take it out of glycogen, and then you have to take it out of the cell. If I were to break one of these bottlenecks, what would be the worst in your mind? Yeah, so that is the correct answer given what I'm about to tell you. Getting it in would also be horrible, like if you actually couldn't get glucose into the cell, because then you just wouldn't be getting energy and you'd have to use fats and stuff. But so yeah, so if you break here where you're getting it out, it doesn't matter anything that's going on in the cell appropriately. In fact, it can be bad because you're building up, you're in a state where you want energy, and then suddenly all this energy that's in the cell can't get out. Okay, and if, like for example, in an organ, in the liver, you might be having all this energy that's about to get out, and then you can't. So we'll work through that. Okay, so we're going to be talking about glycogen storage diseases, GSDs, okay? And the objective ones that I'm requiring you to know are type 1 through type 5. Okay? I'm going to specify that a little more, right? So I'm giving you all of them just so you can see them. These are the only ones I can test on. I'm doing these because type 1 through type 4 is by far the most frequent, okay? Type 5 is neat in respect to those, okay? And it was the next one, so I could group it. And by far for the boards, the one that you would focus on the most is type 1A, and that is also what I'll spend the most time on, okay? So if you're looking at what these breakpoints are, I'm boxing here type 1A, right, as the most important and the bad one. So here is glucose, right, and glucose is coming in. So we're going to be spending a ton of time right here, glucose 6-phosphate, right? And the main disease we're talking about, von Gierke disease, which is type 1A, or type 1, is when you basically can't get out, right? That is blocked, okay? And so if you have a lot of this, everything's going to happen. You're going to go this way. You're going to go this way. You're going to go this way because you're building up this. That's the big take home. We'll hit again and again. All right? And so for all these diseases, what kind of inheritance pattern do you think they'd have? Yeah. Yep. Because it's an enzyme, and if you have half the enzyme, you're okay. I think this actually clears. Okay. So here is von Gierke disease, okay? And so what happens with this, we're going to talk through the clinical presentation, okay? But you're stuck with glucose 6-phosphate. You're not having glucose 6-phosphatase, so you're stuck having the phosphate group on the glucose. So you end up with growth retardation, enlarged liver. You might want to think about why that might be, and kidney. Hypoglycemia, low glucose in the blood, elevated blood lactate, cholesterol, triglycerides, and uric acid. So a lot of those probably shouldn't make sense to you right now, okay? And so we'll get into them. So you've seen these maps from me before. This is the, but not for these exact diseases. These are the what is involved in prenatal screening in different states, right? And so again, Indiana specifically doesn't look good. Blue is what is covered, and we are surrounded by blue for GSD1 alpha and for Pompe, and we're the sole one not doing it. Now, I'd like to thank Dr. Gardner in the back for, he's on some listserv and found that next year we are due, this was passed in March 2019, so starting next summer, we will start having in our prenatal screen Crab disease, Pompe, and Hurler syndrome. We still won't be doing MPS 1. Okay, so the disease I'm talking to you about today will not be part of the prenatal screen. And so I'd point out, for example, Pompe is about 1 in 20,000. So GSD1 alpha is 5 times less frequent. So that seems to make sense. But again, you have to know your patient population. If you're from Ashkenazi Jewish descent, it's about 1 in 20,000. So it's just like Pompe, but they're not being screened, right? Okay. So here's the case. You have a newborn evaluated with failure to thrive. And they clearly have this enlarged abdomen, okay? Blood work reveals hypoglycemia, these things that we mentioned before, hyperlipidemia, high lipids, hyperuricemia, hyperalinemia, high alanine, lactic acidemia, and high cholesterol, okay? So you should feel good if at the end of the lecture you're getting why those are. This is a fixed liver biopsy, so this isn't what the raw tissue would look like, but basically what I want you to get is that in the diseased tissue, it's a fatty liver, okay? There's fatty deposits. So you have the take-home here. I'll just give stuff away because I'm going to repeat it again and again, but the take-home is, has LaFontaine talked to you about the phosphate group yet? No, he normally does like an interpretive dance about, basically, it's this huge, huge thing. When you add a phosphate onto something, think it's like a mega change of I'm charged, and it totally changes the structure. So if you have glucose and you add on this phosphate, it's a totally different molecule. And so when you have glucose phosphate in the cell, that is the way that the cell locks the glucose in the cell. It's not getting pumped out. Okay, and in this disease I'm talking about, you can't take that phosphate off. Okay, so in the fasted state, we'll talk about this again and again. In the fasted state, you're going to want to get glucose out of the liver, right? And so you'll turn on machinery to do that, and then you'll suddenly have glucose-1 phosphate, and you won't be able to get it out. Okay. So, if you did an analysis, you would find you don't have glucose 6-phosphatase. I've already told you it removes that phosphate group, and it's expressed in the liver and the kidney. We're going to be focusing on the kidney, and that's where glycogen is stored. Okay. I feel very bad for you whenever I read some of these clinical presentations. So here is a child with GSD1 alpha, and so unfortunately, they're not going to present to you with this drawing on their abdomen, but what do you think the doctor was drawing here? an enlarged liver, right? And so listen to the presentation of this child, right? This is the diseased child. This is how they're describing them to notice them. They're usually small for their age, which is a child. They have protuberant abdomens and chubby cheeks, child, and spindly arms and legs suggesting a Cupid doll appearance. That all sounds like a baby to me. Somehow you're supposed to kind of see that presentation and think, oh, they're having maybe GSD1. All right. So we know glycogen is the storage form of glucose. When is it synthesized? During what state? Yep, during the fed state. It is degraded when blood glucose is low. In skeletal muscle, when is glycogen degraded? Yep, intact. Basically when you're active. So a student asked me to remind everyone, it's okay if you're shouting something out maybe if I repeat it, but if you ask a question, do it into the mic, because half the class is at home and they're like, I want to hear my colleagues. I said that nicer than they did, but okay. So, right, so there's a big difference there that the skeletal muscle uses glycogen when you need to move. And the liver is degrading glycogen when your whole body needs glucose. And the muscle is a selfish organ. So the glycogen in the muscle cannot be used to maintain your blood glucose. So your brain needs blood, it needs blood, but it also needs glucose. Your brain needs glucose, that's the liver's job. And there's a ton of glycogen in the muscle that unfortunately, or depending on how you think about it, can't be used for the brain. All right, so this is saying that, right? So glycogen in the muscle, using it for energy, glycogen in the liver, using it to pump glucose out into the blood. So the first step, remove the phosphate group. It's not locked in the cell, and people with GSD1 alpha cannot do that. There is a type of GSD1 beta. I'll hit that at the very end, but I'm just talking about GSD1 alpha for now. They're very similar. Okay. And so in the muscle, do people with GSD1 alpha have a problem? What do you think? Right, I see head shaking because they don't want to get the glucose out of the cell, right? I mean, conceptually. All right, so I boxed out everything here, so I just want you to focus on a few things, right? So here's your glucose. You're bringing it into the liver, GLUT2, no problem, right? Someone with GSD1, they can eat a pasta meal, bring in the glucose, not an issue, okay? Glucokinase, they're adding a phosphate group, no problem, right? So they get here, okay? Glycogen synthase is activated. It's dephosphorylated, but it's also activated by this reactant, so that's fine. So they can make glycogen, right? And they can also go down and store that energy by making acetyl-CoA and making fat, right? Check. So they can do all of that, okay? So basically, think about people with this disease as, I don't think we have this in this city, but there's some bigger cities where you have highways where a lane can change during traffic, right? So there's 3 lanes going this way in the morning and then 3 lanes back in the evening. Your metabolism's kind of like that, where like you're shoveling arrows in one direction depending on your need. This disease is like the entrance is always open. You can always take in energy. Glucose is always going, but the exit ramp's always closed. So traffic problem, right? Okay. So the liver traps actually over 60% of the meal. I am always surprised how much the kidney comes into play in a fast. When you're fasting, the kidney can be like 40%. But basically remember, your liver is your Tesla battery where you're storing that sugar energy. All right, so let's talk about the fed state, okay? In the fed state, and we're just talking about normal, everyone, what's going on? Is glucose phosphorylation up or down? Let's see the Roman emperor, right? Up, good, I saw up. Glycogen synthesis, up or down? Very good, up. Pentose phosphate pathway, up or down? Yep, up. Glycolysis, up or down? I see sideways thumbs. Up, okay, we'll get into that. Gluconeogenesis, up or down? Yep, got it, down, very good, switch it up. Fat biosynthesis, up or down? Up, yep, you guys got it right, okay? So basically, you are storing energy, right? You are building up this, and so you can store energy as glycogen, you can go down, store energy as fatty acids. This is a reactant, you can go over to the pentose phosphate pathway, okay? You are not trying to do gluconeogenesis. You don't need more glucose, right? That you don't need in the fed state. Okay. Everyone good on what I've said so far? How's the speed? Good, too fast, too slow? Too fast or good? Good, okay. All right. So here's that big map again. The only thing not really shown here that I want to highlight is that if you're in a high energy state, you're shutting down the TCA cycle. So you guys are used to ATP being high energy, right? But I've also now talked to you about NADH being a marker of high energy, right? So in both of those, you're blocking this, and so you can come down to acetyl-CoA in a fed state, and you can go off and make fats, right? So just remember NADH, high energy. I've shown you this graph before, correct? Yes, I showed this a few times. I love this graph. I will point out that this is a generic, like these lines kind of shift with the individual and where you are. But what's happening in the fasted state? So at 6 p.m., let's say you have a meal, your last meal for the night, because I've taught you not to eat late at night to help your circadian rhythm. 6 p.m. you have dinner, okay? So where is your glucose coming from? Your glucose is coming from the meal you just had. So for a while, you're not using anything except what you ate, right? Then you go to bed. By 2 a.m., you've started to use your glycogen, right? Because you're starting to enter a fasted state, okay? By 10 a.m., your glycogen is starting to go down, and you can start to do more gluconeogenesis, okay? And then by 6 p.m. the next day, about 24 hours, then you're kind of in gluconeogenesis realm. Now, I will point out that this blue and green line, I think can shift to the right a bit from that. Like if you have been eating a normal Western diet and like going to Olive Garden, your glycogen store can be pretty big, and so it can last longer. And so sometimes if people are trying to like get into ketosis, if they're using like urine strips, that won't happen here where you think it might be happening, right? It might happen a little later. Okay, and we can talk about ketosis more later on. People bundle a lot of things. There's a lot of things happening, right? If you use a ketone strip, you're measuring ketosis, but you're not necessarily measuring autophagy, which is another great thing I've talked to you about. So those are slightly different, but they tend to happen temporally in the same time. Okay, so glycogen degradation in the liver keeps your blood glucose up. And here I'm color coding what's happening with these lines here. So last night I added this down arrow. I was trying to point out that basically when you have glucose 6-phosphate in the fed state, you're making glycogen, right? So this arrow is this arrow. But you're also, I added this because you are also in the fed state going to make fats, right? So then after that, then you start to enter using your glycogen. So you're coming the other way down, right? Getting the glycogen. I'm not going to keep toggling these things. Okay, then you start to do gluconeogenesis, right? You're starting to make glucose. Let me get out. Okay. All right, so where are the fats coming from during the fasted state? Super, super softball. They're coming from your fat, from your adipose tissue, right? Yeah. So that is your energy store when you're out of your sugar warehouse, right? So your liver kind of stores your glycogen, which is like your sugar Tesla battery, and then your adipose tissue is your liver Tesla battery. Okay. So these are released as ketone bodies into the blood, and that can cause slight acidification, which we talked about in the presentation, right? Where do the amino acids come from? They come from muscle, okay? And they come from all cells of the muscle. So there's not a mechanism to really control when you're in a fasted state where to take amino acids from. And so most of your protein weight is in skeletal muscle, right? So you can degrade skeletal muscle when you fast for a long time if you're trying to lose weight, for example. But you can take some from your heart, which is also a muscle, which is not ideal, right? But it's kind of a ratio thing where that's not by weight most of your muscle. Okay. The only thing, and I think I've told you all of this before that I want you to see here, is this part again. So we've been talking about fed and fasted state. So for caloric restriction, what is happening? You're increasing that good thing autophagy, you're decreasing mTOR, which is good, and then you're increasing senescence so you can kill off cancer cells through caloric restriction, and you're decreasing inflammation. High level good things in the fasted state. This picture you already had on another slide, okay? I'm just highlighting here. I think in the last slide, I actually underlined them, so you probably already got this, right? So you have the GLUT2, you have the glucokinase, you have the glucogen synthase. You can go and activate the pentose phosphate pathway. I didn't mention the pyruvate dehydrogenase, but basically all these things are on in people with GSD1 alpha, right? So with me underlining the last slide, you really didn't need that. That was all repeat. Okay, so let's now talk about this disease state, okay, in the fasted state. What happens if the child is in a fast? They can't use this blue line. They can't use the glycogen, right? I've talked about already this bottleneck, right? Can gluconeogenesis help them in a fasted state? Now we're on to the green line. Yeah, no, why not? That's right. Not into the mic, but well done. They can't. What she said was they can't get the last phosphate off. So even if they make the glucose, they can't get it out. So if you think of your liver as, your bus terminal central of making glucose, you're getting all the signals to make the glucose, but then it's the buses are piling up there. So this is why you have the enlarged liver, okay? You have all this glycogen that turns into glucose 6-phosphate. Depending on your disease, you might branch off some of it, but it's stuck there. You can't get it out. So they can have profound hypoglycemia in the fast, right? This can be lethal because they don't have sugar in their blood. So these are not numbers you have to memorize, but rough magnitudes that are something to have a good idea of. So where is glycogen stored? What is that magnitude? So in the liver, you have 70 to 100 grams. The muscle has 400 grams, so 4 times as much. But again, that is a resource that your blood cannot use. That is for, my wife hates me saying this, but to run away, from lions on the African safari, right? Like we've evolved to, we need our muscles to move. It's reserved for that purpose. Right, so your carbs are gone in about 24 hours. I've mentioned this again, but I keep driving it home that as you lose a gram of glycogen, you lose 3 grams of water. So as people diet, they lose a lot of weight and then they'll rebound and get depressed. So, just have them track a moving average. It helps them maintain their goal weight more. All right. So we can explain hypoglycemia, right? Because they can't get glucose out of the liver. Okay. And so you're building up this glucose 6-phosphate, but it doesn't just stay there forever. It follows all the arrows, okay? So let's start following them, and as we follow them, we'll find different clinical presentations. And this part will start to go pretty fast, but that's because it's pretty simple. You just follow the arrow, there's a buildup there. How does that describe that clinical presentation, okay? Okay, so hypoglycemia, we've got check. How about lactic acidemia, right? Can anyone potentially see that? Actually, it's all shown on your slide, isn't it? I think it is. Right. So, boom. If you look in here, as you build up glucose 6-phosphate, I don't even have to draw. The arrow's there for me. Right? You're going down, and when I zoom in on this region, when you go down and get more pyruvate, you go over and get lactate. Okay? And this step is done to regenerate the NAD+. You need NAD+ to keep this process going, and so this is a process to regenerate that from the NADH, okay? And so you end up with lactic acidemia. All right. How about the elevated triacylglycerols? You are making more fats, right? You're also going down here, and then this is shut off down here, so you're going up and making fats. Just following the arrows. How about the pentose phosphate pathway? The big kind of gloss over there is that this is involved in making bases for DNA. So what happens to extra nucleotides because you have glucose 6-phosphate building up, so you're going into this pathway? Well, what happens is you have a buildup. You don't need to know any of this stuff, but you end up down here with uric acid, which creates urate, and chronic hyperuricemia can lead to, can anyone tell me what's underneath this without removing it? Gout. Yes, very good. The shout out instead of the microphone, gout. Very good. So here's a picture. Here are shown crystals, right? So they crystallize and cause pain. Another clinical presentation. People, I don't want you guys to delve into too much detail. What you need to know there is that this buildup of glucose 6-phosphate turns on the pentose phosphate pathway, and that causes increased uric acid, which leads to hyperuricemia. Okay? All right. How about hyperalinemia? I don't kind of expect you guys to remember this, but how does the body move protein around from the muscles? It does it through alanine. So you end up having high levels of alanine in the blood because you're not having any glucose, okay? And so what is the treatment here? I meant to ask you this before I showed you this slide. The treatment is basically these children cannot maintain a blood glucose level. So if it was non-invasive, you would have them walk around with a drip that just kept them at a normal ratio, normal blood glucose. But really what you want to do is give them something that's like a slow-release drug. And so a very cheap, viable thing that works is raw cornstarch, right? So you just want something that has a glycemic index that's a slow release. So like powdered sugar, not good for them because they'd have a pulse of, okay, I'm okay, but then they'd crash out because that glucose gets sucked up, their on-ramp's fine, and then they can't get it out. Everyone good? Okay. And if you mess up, if they're not taking the raw cornstarch, it's bad in both directions, right? If they don't take it, then you're not treating them. So they have hypoglycemia, right? Low glucose, hyperlactidemia, all these things we talked about, hyperuricemia, retarded growth. If they take it a lot, then they can have glycogen overload because you're still giving them way too much sugar and it fills up their liver, okay? So this is again a case where gene therapy could be great. In particular, it's good that the two things to look for gene therapy are Is it a single gene? People won't even talk about it if it's more than one gene, like diabetes is complex, right? And then where do you need to get it? And this is one case for GSD1 alpha. If you got the gene in the liver, you'd pretty much be good, right? You don't need to worry about getting it into the brain, into the muscle, right? Okay, so let's compare that to GSD5, myocardial disease, okay? And I'll show you I have a summary slide of all of these in the next 2 slides, I think. So the take home here are, think about muscle for myocardial, okay, M's. And so basically you're again having a problem breaking down glycogen, but it's not as big an issue because it's in the muscle. Dr. LaFontaine may talk to you about this because he used to play soccer with someone that had this. And so these people sometimes will like take sugar before an activity so they can kind of get through the time frame where they don't have sugar to move. Okay, so here's basically the summary slide of these diseases. I covered 1, 2, 3, and 5 here. So we already talked about how it's recessive. So von Gierke disease, the 1, 2, and 3 are liver manifested. Myocardial is muscle, okay? So here are the enzymes. I won't read through them that are down in each of them that are not working. The findings, we've already talked about von Gierke disease, right? Increased glycogen in the muscle. These are all obviously going to be bad. Increased lactate, triacylglycerides, and gout. And the notes are that you want cornstarch is a good oral way to treat these kids. You would avoid fructose and galactose, and decreased gluconeogenesis and glycogenolysis. So for Pompe, they have cardiomegaly, hypertrophic cardiomyopathy, the problems with the heart, hypotonia, exercise intolerance, and early death, and they're affecting the heart, liver, and muscle. For Corey disease, this is like a milder version of type 1, and so this is because it's due to the debranching enzyme. What happens is they kind of break off parts of glycogen, and so you end up with this thing called limit dextrin, which is like a piece of glycogen in the cytosol, okay? And so if you're looking at like a differential compared to GSD1, they would have normal blood lactate, okay? So for myocardial, the big thing there is that it's muscle, right? And it goes with muscle. I think I took this mnemonic from first aid, very poor carb metabolism for these 4, which I liked, okay? And so I talked to you already about how they have this second wind with increased blood flow, so they have to basically dose themselves with sugar if they're about to do an activity. And so again, as a differential, they would have normal blood glucose levels, okay? So it's very kind of good to hone in on differentials when we're talking about a class of diseases. This slide is all a review of the last one. I cleaned up that slide last night, so I think the one above is better. You don't need to know any extra information. There probably is some extra information here, but you don't need to know it. And so now to quickly go through the difference with GSD1 beta. So here we're showing what happens to glucose 6-phosphate. And I glossed over this before. I mean, I actually just didn't tell you. But the way that the phosphate's actually taken off is a 2-part process. So at first, it gets transported into the ER via this G6P transporter. And then it has this enzyme that takes off the phosphate group. So most of the GSD1s, 80%, are alpha, GSD1 alpha. And that's the problem with the enzyme that's actually taking off the phosphate group. GSD1 beta, the problem is with the transporter, okay? Now you'd think, okay, those should be identical because in the end, you're having the same problem. The reason GSD1 beta is worse is because it's more ubiquitous. Okay, so it's not just the liver. And so you ended up with GSD1 beta, you ended up with poor wound healing and increased infections. Your white blood cells actually die because of metabolic disorder inside those cells. Okay. And so most people wouldn't do this, but to quickly show you kind of a Venn diagram of how things can present similarly and differently, I'm going to compare GSD1 alpha to diabetes, and normally those would be thought of as exact opposites, because GSD1 alpha is I can't get sugar into the blood, low blood, right? Blood glucose. Whereas type 2 diabetes is we can't respond to insulin enough. There's too much sugar in the blood, hence glycation, right? So I'm going to show you GSD1 alpha up here, diabetes down here, and everything in red is different between them, and everything in yellow-orange is similar. So in GSD1 alpha, you are having this increase in G6 phosphate, right? You end up with decreased glucose, high hypoglycemia, so those are different, but you do end up with increased free fatty acids, which is the same. And you end up with steatosis and lipogenesis. All of those are the same. These other things are different in GSD1 alpha, right? An enlarged liver, clearly, because the stuff's getting trapped in there, and increased lactate outside. So down here for diabetes, obviously, decreased insulin sensitivity, increased glucose, those are different things. But I wanted to point out how you'll see presentations in blood work, and there's some overlaps, and the overlaps with the differences help you with that differential, right? So, pretty good timing, like 4 minutes over. As a take home, here's the kind of big summary again of where things are going. You're storing your fat here, you're storing your sugar here, and so these are your energy warehouses. I'll talk to you about storing protein in a future lecture. I'll kind of mention it now. This is actually my last foundations lecture to you, so I'll see you in CPR again and then more in the second year. So you got a bunch of me and now I'm out. But, so I'll ask you a few kind of roundup questions here while I talk this through. So go ahead and answer this while I talk. Protein are a unique case of energy storage, right? Glycogen, clearly it's our sugar storage, no problem, that makes sense. Glycerol, that's its only purpose, right? It packs, it stores fat, it stores energy. But proteins are our nanomachines that do a lot of stuff. So we'll hit this in GEM in the GI course next year. But when you are in a fed state, right, you have kind of reagents to make your nanomachines. So you can make proteins, you can make muscle, right? And when you're in a fasted state, you can use that muscle for energy, but then you're degrading, things that you need. So it's not, when we start to talk into things that move our metabolism, let me just see where we're at here and we'll move on. Okay, I'll look at that later. And then answer this one, online notes. It won't always make sense for amino acids. We can't anthropomorphize them as well because it can't be like we evolved to do this because there's just conflicting signals. There's times where ideally we would want energy and to keep our muscle, but we just seem to, we can't do that. Okay, that is it. I'll have the whole lecture thing after this, but any questions for the audience? Yes.
So why is it always the Ashkenazi Jews that kind of get?
Yeah. So there's a few reasons. One, they are just any population that is more inbred or, they're genetically unique, right? So that's one thing. I would guess that there's probably an affluence factor that they, I'm sure there's subpopulations like of Native Americans that haven't been studied as well that are prone to things. So I don't think it's a justifiable reason, but the scientific reason is that they shared their gene pool and were not infiltrated by other genes, right? Okay, let's see this. Okay, and then just let me know how the lecture is and then come on up with any individual questions.
So looking at the Lineweaver-Burke plots for non-competitive inhibition.
Yeah, for these I'm gonna hold on. Let me close this.
Okay.